Hello class, this is Mr. Mormon. This is going to be a review for our upcoming test on the chemistry of life. The test is going to focus mostly on the information that we've done more recently. That would be organic molecules and enzymes, the second part of chapter 2. But it will also cover some other things that I will go over right now. This is meant to be a brief overview. Remember, you can always go back and look at earlier videos that cover specific things. There's a whole video on enzymes. There's a two-part video on organic molecules, all of which would be good to go over if you're unsure of any of the things that you see in this video. Okay, let's start with organic molecules. There's four basic classifications of organic molecules that we talked about in class. There's carbohydrates, proteins, nucleic acids, and lipids. You should know the main function of each one, be able to give examples of each one. Remember, we have our study cards. You can go back and review those or go back to the video on organic molecules. Remember what makes an organic molecule an organic molecule is that it will have both carbons and hydrogens in it, and usually there's going to be carbon-to-carbon bonds as well. Remember that carbon is very useful because it can make so many different molecules. It can do this because it makes a lot of bonds. It makes 4 bonds, which is more than most elements can do. It can bond with lots of different things: hydrogen, oxygen, nitrogen, phosphorus, and other carbons, just to name a few. The bonds can be single, double, or even triple. The carbons, when they link together, can form long chains and even rings. Now let's talk about a special group of organic molecules, the enzymes. These are some of the specific things that you should know about enzymes. They're biological catalysts. Remember, a catalyst is a chemical that speeds up other chemical reactions. And enzymes are very important because they're basically keeping all living things alive. All living things have a metabolism, which is the sum of all of the chemical activity inside, and that's all being controlled by enzymes. You should remember that enzymes are proteins, and we could also say that they're polypeptides. A polypeptide and a protein are the same thing. They're not used up in chemical reactions. They get recycled and can do a reaction over and over and over again. Remember that enzymes are specific. They can only do one job. Remember the demonstration we did in class with the salivary amylase. Right in our mouth, the amylase dissolved the starch very easily from the cracker, but that wouldn't work on a protein. In the same way, the pepsin, which is in our stomach to dissolve proteins, would do nothing to a starch. Remember that enzymes work because of their special shape. Remember the lock and key model. I'll show you some examples of that. Remember that they also have an optimal pH and temperature. This means that there's a temperature and a pH that is specific for each enzyme, but it's the pH and temperature where they will work best. If you get higher or lower pHs or temperatures, they will not work as well. This diagram is showing how an enzyme works. This large purple thing is the enzyme. Notice that it has an activation site. It's got a special shape that will accept the substrate. This is the molecules that it's going to work on. Here we see the molecule binding to the activation site, creating the substrate, the enzyme substrate complex. The chemical reaction happens because now there's stresses added to the bond that was between these two molecules, and they're broken apart. This is sucrose. This is basically table sugar, and it's being digested here. This is a digestion reaction where a larger molecule is being broken into 2 smaller molecules, glucose and fructose. This next one would be an example of a synthesis reaction. Here, the substrates, my 2 reactants, are combining with the special shape of the enzyme, and they're being lined up just right so that a new bond can form. And this is a synthesis because we started with 2 smaller molecules and ended up with a larger molecule. Now these diagrams are pointing out, these graphs are showing what I was talking about with the optimal temperature and pH. Here with this enzyme, we can see that it's going to work best right around 38 degrees Celsius, 37 degrees Celsius. That's actually body temperature. So this might be an enzyme from the human body. At 37 degrees Celsius, it's going to work the best. If you cool it down or heat it up, notice that it's not going to work as well. Same thing for pH. This one seems to like a pH that's slightly above neutral, so that would be slightly basic or alkaline. This idea is important that we've talked about quite a bit, that many of the organic molecules are formed by polymerization. And what that means is you have small molecules that are chained together or linked together to make these long, long chains, which are called polymers. Now, there's three that you absolutely have to know. You're going to see this on your Regents exam, and you'll certainly see this on this test. You need to know that sugars are the building blocks of starches. They're the monomers that build starches. And remember, both of these would be carbohydrates. Remember that amino acids are the building blocks of proteins or polypeptides. We saw this in the lab with the amino acids that we strung together to make the dipeptide and then the polypeptide. We were on our way to, sorry about that. We were on our way to building a protein. So amino acids build proteins. And nucleotides are the building blocks of nucleic acids. Another thing to remember, you could go the other way and say that when a protein is digested, it's broken down into amino acids. Or when a starch is digested, it's broken down into its monomers, which would be sugars. Okay, let's talk a little bit about nucleic acids. Here we see the basic structure of DNA. It looks kind of like a ladder, which is twisted. You should know that nucleic acids in general, they store and transmit genetic or hereditary information. And what that really means is that it contains the information on how the amino acids are put together to build a protein or polypeptide. It's the DNA which acts like a blueprint, the instructions on how to build all the proteins that are in your body. The protein that gives you your eye color, your hair color, those are all, it's in your DNA, and that information can be passed from generation to generation. So that's what we mean by hereditary information. You should know that the building blocks would be nucleotides. And these diagrams show that a little bit better. This would be a small segment of DNA. You can see that it's double-stranded. And each one of these segments here would be a nucleotide. And here's the structure of just one nucleotide. There's a phosphate sugar. And you can see going down the spine here of the DNA molecule, we've got phosphate, 5-carbon sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And we've got the same thing on the other side. Now, this is where the actual information is stored, here in the middle with these nitrogenous bases. And there are 4 different ones: adenine, guanine, cytosine, and thymines, A, T, G, and C. And there is a rule we're going to learn when we get more into DNA, that the A's always have to combine with T's and G's always have to combine with C's. Okay, here's something else that we spent some time talking about, what pH is, and the pH scale. The pH scale goes from 1 to 14, with 7, which is neutral, being right in the middle. Pure water would be neutral. The lower number are acids. Remember, the farther you get away from neutral, meaning the lower the number, the stronger the acid. And going above neutral, those would be basic or alkaline. You can see some of the different things that fall, where they fall on the pH scale. Again, the higher you go, the stronger the base. Okay, the farther away from neutral, the stronger the acid or base. Next, we need to really understand some of these ideas that we talked about, the difference between autotrophic nutrition and heterotrophic nutrition. Remember, autotrophs are self-feeders. They're making their own food. Heterotrophs, they need to eat and digest their food. That's how they get the energy they need to eat. And if I've kind of diagrammed the two different types of nutrition down below here. This would be photosynthesis, which is how most autotrophs make their food, and the food is glucose. The energy that they capture from sunlight is stored in the chemical bonds of the glucose molecule. Then in cellular respiration, that energy gets released. Glucose and oxygen go into the cellular respiration equation. The glucose gets broken up into carbon dioxide and water, and energy is released from all those bonds. Now, it's very interesting to notice here that they're just totally exact opposites, these two reactions. The things that are the reactants in the photosynthesis reaction are the products over here in the cellular respiration reaction. And the same thing, the glucose and oxygen are produced by photosynthesis, and those are the exact things that you need for cellular respiration. Another thing to note is that autotrophs that do photosynthesis will also do cellular respiration. The only difference is that they are actually making their own glucose first, whereas heterotrophs will be eating starches or sugars to get the glucose. They eat carbohydrates. Okay, just a few more things that I'd like to review here. Remember the phrase dry mix? That's how we remember which variable goes on which axis when we're graphing. DRY stands for dependent and responding variable. That goes on the y-axis. MIX, the manipulated or independent variable, goes on the x-axis. Okay, and now for some reminders about measuring. When measuring mass, we're going to use a triple beam balance. We should always measure and record in grams when using the triple beam balance, and measure to the nearest tenth of a gram. Volume is, if you're measuring liquids, you'd use a graduated cylinder. You're going to measure and record in milliliters. And remember to record the measurement that you see at the bottom of the meniscus. The meniscus is this curved shape that you see because of adhesion. The water gets pulled up the sides of the graduated cylinder. For measuring the volume of regular solids, you're going to use a ruler and the proper formula. For example, a cube, it's going to be the length times the width times the height, or a rectangular prism, same thing. Measure in cubic centimeters. That's what we're going to be using for when you do these calculations, you'll end up with cubic centimeters. And also remember that a cubic centimeter and a milliliter are actually the same volume. If you need to find the volume of an irregular solid, you can use the displacement method. If it fits in a graduated cylinder, you can do it like it shows in this diagram. Otherwise, you could use a larger container that you fill to the very top, put your object in, and catch the water that spills out in an even larger container. The volume of that water will equal the volume of your object. All right, well, that's a pretty good review of most of the things that are going to be on this test. Remember, go back and look at old videos if you need more help. And I wish you good luck and good studying. And we will see you for a review session just before the test. And you can ask me any further questions that you have then. Okay, see you tomorrow.